we use this female nude mice that has no thymus, and so to which the gastrointestinal stromal tumor has been orthotopically induced in the xenograph, and so you see comparable activity in terms of anti-cancer, anti-tumor activity for both. And so the untreated mice, you see the exponential growth of the tumor mass. And so, but then when you look at the cardiotoxicity, the golden indicator, the golden parameter here is the percentage of left ventricular ejection fraction. And so a 10% decrease is, of course, lethal because it implies a massive dysfunctionality in the heart. And our compound, if nothing else, retains the same ejection fraction as the controls, if not improves it. And that may be due to the protective role of anti-JUNK activity on the heart. So here is a much more convincing picture, perhaps. So here is the MRI of the heart of the mice treated with imatinib and WBC4. This is in the xenograph study with the nude mice, xenograph GIST study, gastrointestinal stromal tumor model, and so you see the left ventricle in the diastole and in the systole. If nothing else, the cross-section almost remains the same and is even compensated by a bloated auricle. You see, this heart is really not pumping well at all. In fact, these mice had to be sacrificed, or actually died after the 6 weeks of treatment. Whereas with our compound, that remember only differs in 1-methyl from this one, these mice it's perfectly fine, right? I mean, you see how tiny the cross-section of the left ventricle is in the systole, right? So this is actually a functional well-pumping heart, and so I think I've tried to show you in these slides that using the wrapping concept, you can actually distinguish paralogs in ways that will enable you to control specificity so that you can curb side effects by telling apart paralogs. So you may say, yes, well, that's an example, but is this universal? So to what extent can we control specificity by using this wrapping concept, right? I mean, it may be that this was a success story, but just a product of luck rather than anything else. So I decided I would just compare the wrapping patterns of major drug targets with the with the pharmacological profile of those targets. So you could actually compute what I like to call the wrapping hull of a protein. So the wrapping hull are all the dehydrons that are actually wrapped by a drug. So here is a drug, here is the dehydron pattern of the drug, and those are the dehydrons that are affected by the drug, right? That the drug, where the drug is making a difference in terms of enhancing the local stability of those vulnerabilities. And then you can actually align those hulls, because these are close paralogs, and look at the wrapping differences, right, across paralogs. So how do you look at, what do you mean by wrapping distance? Well, so for example, remember the dehydron pattern can be encoded in a matrix, right, in a binary matrix. 1 at position ij, if I and j are paired by a dehydron, 0 otherwise. And then now that each protein is a matrix, you can actually take the Hamming distance of those matrices, and we call that the wrapping distance, right? I mean, perfectly fine, because if the structures are alignable, then the matrices are alignable, and if they're not, you can use you have to tinker a little bit with them to compute those Hamming distances, but in principle you can do it. And these are all targets that have structure in the PDB, and these are the wrapping distance matrix across those targets. for all those pairs, right? Those pairs of protein targets. And then you can independently look at the pharmacological distance matrix. So for each protein, you have your affinity profile against a background of drugs that are successful. So here, we have to exclude staurosporine, which is a highly promiscuous compound. It's not really an effective drug at all. And so for each protein, you can associate to it a vector, and then you can take the Euclidean distance of the affinity vectors, right? And so that would give you an idea of the difference in pharmacology across between two proteins, right? So I mean, the distance would be 0 if the two proteins react identically to a background set of drugs, and one in the opposite case, in the other extreme. And so you can define actually a pharmacological distance matrix. And then the question is, okay, how do the two of them compare? The two of them compare. And they actually correlate alarmingly well. Although none of those drugs were designed using the wrapping concept. I mean, if they were designed using the wrapping concept, then it would be completely tautological, and the correlation would be absurd, right? What I'm saying here is that these drugs that were found through trial and error and painstaking effort actually are essentially obeying the wrapping concept, although they were never meant to be designed using the wrapping concept. And so now that we know what makes a drug successful, we may use it rationally and intentionally to create better drugs, right? Because, as I said, none of those drugs had the wrapping technology in mind. But this correlation best speaks of the apparent universality of the concept in terms of trying to develop a new filter to achieve specificity based on structure-derived features of the purported targets. And with that, I would like to end and thank my funding agencies and my group that did all the work, and all these people that helped me think straight about these concepts. And also I want to thank you for your very kind attention.
Questions?
Yes.
Does any of your target protein has a crystal water into that one?
Crystal water, you said?
Because sometimes the water is also bridging between the backbone hydrogen bonding, and if you are bringing the hydrophobic, then it disturbs the water, and how does, at that time, the wrapping technique is? Yeah, so, well, that points to a very important problem, what you're saying, which is normally these dehydrons are in the twilight zone between order and disorder. So water really they are induced folding features. So in other words, if you don't have a ligand, chances are water will get in and disrupt these bonds. So oftentimes what we see is if you have a poor wrapper, or and yet in a crystal structure you see water intercalating oftentimes. The idea is that we don't really know exactly. I mean, this is a very dynamic object. In other words, when we say it's very underwrapped or it's vulnerable to water attack, what we are saying is essentially we're introducing some underlying dynamics into the picture, right? We are saying, in the absence of a ligand, if you examine that particular paralog, chances are that that hydration of the backbone will prevail. But given the right ligand, that hydration is hindered and the structure is retained. So we are, in a way, this is pointing to the problem of, what constitutes the folding, the quality of a particular fold. Is it, when we identify those interactions, those packing defects or wrapping defects, if you wish, we are actually, that seems to be indicative of ephemeral, adventitious, or permanent complexation. So the structure has to be viewed in the context of some level of complexation. In the absence of that level of complexation, I don't think you really have locally a structure there. It's a again, you can actually show that because you can infer dehydrons directly from sequence. Because from sequence there's something you can infer with a good degree of reliability, and that is the propensity for disorder, the disorder propensity, right? Now there are algorithms to infer disorder propensity, and we don't care about regions of certainty of disorder, but we care about regions that are in between order and disorder. And you can see the disorder profile across the sequence, and what matters are the regions with high slope, because those are regions of tamed disorder, regions where it may be disorder, but it's not a wild disorder. If you have the right ligand, you can actually maintain the structure. Alternatively, regions that lack tertiary structure, for example, a protruding piece of say, the p53 DNA binding domain, are also very rich in dehydrons. In other words, if there's no ligand, water will just come in and hydrate the backbone and dismantle the structure. Also of significance is proteins that really cannot keep the structure at all. For example, amyloid proteins. So when you ask yourself, well, what is the most underwrapped protein of all in the PDB? Well, it's a cellular prion structure, the cellular structure of the prion. In other words, it hardly qualifies as a soluble protein. It's so vulnerable that it hardly qualifies as a soluble protein. What most likely happens is it aggregates, it goes into a different phase. It really cannot hold its monomeric structure at all. Other highly underwrapped proteins are toxins, for example, but they hold the structure because they have a lot of disulfide bridges. So it's a very dynamic concept where water has a high propensity to get in and dismantle.
Have you done the corresponding experiment, and I realize it's non-trivial, where a methyl group could fit? Because obviously if it doesn't fit, it's not going to bind. But in a region which you would predict not to have a different wrapping state, and show that doesn't modulate the binding specificity? So you basically take a methyl group, let's say there's a hole over here. Okay. Here one guy is wrapped, here one guy is underwrapped. Here the wrapping state is exactly the same, but there's a hole in the structure. So I could shove a methyl group in there.
Yes.
If your hypothesis is right, then in the former case you should see the differential binding affinities, whereas in the latter you would not.
Yes, indeed, yes, we have done those controls. Yes, we have done them with imatinib. That's a very good question. I think there's a hole. There are several holes in here where there's water. I believe it's in this region. So we have it's in this region. So we have expanded. We have expanded the tail of imatinib in regions where there is where there's crystallographic water, but not a dehydron there, a hole in the structure, as it were, and we don't see any difference in affinity. Yes, we have done those controls.
And if you look at the binding sites of between two different proteins, do you see other different, because obviously what you're trying to do is to design specificity, there has to be a physical chemical difference between X versus Y.
Yes.
Are there other differences besides wrapping distances in these binding pockets that you could take advantage of?
There are other differences in, well, there are there's, people have tried other ways of engineering specificity based on, for example, orientation of this highly conserved phenylalanine, for example. And this is the so-called, this is the activation loop. And so they have tried to look at differences in the conformation of the activation loop, for example, which distinguish kinases in the active state, or in the inactive, excuse me, in the inactive state. In the active state, they all look alike, but in the inactive state, there are differences. And yes, they can engineer specificity to some degree, but not in a systematic manner. In other words, it's not. It's just a feature that may distinguish two particular kinases, and there are many others. But not. You can't just have a universe of targets and say I want to work here, here, and I don't want to go there and there, in a universal manner. But there sure are differences in I mean, the region that is more prone to disorder is the region that is also more prone to variability. But it's also the most elusive region because it's more fluxional. And so chances are you're going to induce folding there. And that's a wild problem, as difficult as the protein folding problem. I mean, to try to predict the fold or the induced fold, right? Or where is it going to go? So
maybe this is related to that, but if you were looking at this from the perspective of cancer, how could you predict how to escape your inhibitor? And how difficult would that be?
I cannot predict how it will escape the inhibitor. I know, and I can rationalize a posteriori how it escaped the inhibitor, but I cannot predict it. So, for example, to this wrapping, I don't know what we will do to these wrapping things. Probably we'll modify. We haven't looked at that, but we looked at how it escaped from the parental drag. And sometimes the escape from the parental drag introduces mutations that produce new dehydrons. And then we can take advantage of that wrapping difference in the target to chase the nature in that we can actually create a drug that has affinity for the moving target, for the imatinib resistant variant. Now, it would be interesting to see how nature responds to the wrapping design. In other words, how does it escape? For example, if you target, we have tried that. We have tried to target this dehydron in the activation loop, and by putting a methyl here, and we are asking ourselves, well, what kind of somatic mutations will ever arise here? Probably mutations that will wrap this properly and make it disappear. Or maybe something that even destabilizes the loop further so that this doesn't form at all. Right? So you don't even have so it's so underwrapped, this region, that now you don't even have a dehydron in the drug-resistant mutant. But we don't know yet because the lag time for those experiments it's on a different scale. So it would be interesting to test what are the nature's strategies to escape wrapping designs. Yes.
Do you have any crystal structure that complex with your design drug?
Yes, we do. In particular, we got this one. No, where is it? Just a minute. This is the most striking one. So we got, you see, Junke doesn't even form a dehydron. But if you bring enough grease to the surroundings, you can induce it. And now we make the crystal structure for that. And the idea was to show that induced folding, if it's controllable, in other words, if it occurs in a restricted area, I mean, I don't talk about, induce folding an entire protein, but if you can induce dehydron in a loop, say,
you can really use that as a selectivity triggering feature if you do it properly and understand the rules of the game, right? So here what we are doing is we're just trying to wrap it a little better so that it can actually form. And that was initially shown in simulation, and then crystal structure revealed that the loop closes up. But we don't have, for example, the crystal structure of imatinib, of our wrapping version of imatinib with the targets, unfortunately. It just takes forever. Sometimes it doesn't work. Or yes?
I have a question regarding the concentration of the drug. So the doses of treatment, is it somewhat sensitive? So if you increase the newly developed, the concentration of the newly developed drug, does it then lose its sensitivity or specificity?
We use exactly the same concentration in this experiment than we use for imatinib. Exactly the same, which is 5 milligrams per kilogram or something like that. And so these assays are made at exactly the same concentration. And so you see the tumor is taking off in the xenograph, and this is what's so these experiments. So for example, if you use the same concentration as imatinib, the mice died in 6 weeks. These mice live for about 13 or 14 weeks, and which seems to be their life cycle for nude mice. So, in fact, they were okay, as far as we can see. Yes, sir.
I was wondering if you looked at dehydrons either in nucleic acids, RNAs, or nucleic acid protein. Have you been entirely within protein?
Yes. Well, very important. Well, for example, if you look at protein, dehydrons are particularly prevalent in nucleic acid protein interfaces. And the reason for that seems to be, this is a protein, this is a region of a protein which interacts with the DNA, right? And this is full of dehydrons. And if you look at transcription factors, they're full of dehydron. The reason seems to be that they promote dehydration. And that's exactly what you need to get the right, to get the electrostatics between the DNA and the arginines of the protein going. In other words, not only you need the electrostatic matching, but you need a reason to exclude the water from the interface to get it going. So this sort of microcavity, water can get there, and actually, you see, if the arginines were planted in a flat surface, then they would not be as effective, or lysines, say, because they will be hydrated. But if they are decorating a region where water wants to get out, then that's much better as far as enhancing the electrostatics of the recognition of the phosphates in the major groove.
I was wondering if you'd actually look for dehydrons in the nucleic acid.
Oh, in the nucleic acid itself.
Or in complex with complex.
Well, that's something we haven't explored, but maybe it points to the base stacking issues, right? I mean, why do you have higher stability when you have things that are stacked? Well, you can argue many ways, and there's been a whole, there's a whole theory of that by Sim and all these people, but one argument is that they protect each other, right? They exclude water from one another, and so stacking actually enhances the electrostatics. But we haven't really explored dehydrogen nucleic, but certainly we have examined DNA protein interfaces, and those are extremely rich in dehydrons.
Thank you very much.
Thank you.